Hi, everyone, and welcome to a new episode of our podcast. My name is Sadi Anwar, and I am Acquisition Editor at River Publishers. I'm really excited about today's conversation because we are celebrating an important recognition in the field of computational biology. With me today is Professor Andrea Tangherloni, Assistant Professor at Bocconi University in Italy, and he is also Editor in Chief of the upcoming River Rapid series in computational biology. We will be talking about a research paper that recently received the Best Paper Award 2024 at the 21st IEEE Conference on Computational Intelligence in Bioinformatics and Computational Biology. The paper is titled as Towards Explainability of Dimension Reduction Plots of Unsupervised Learning Model Outcomes. And this paper was authored by Tony Enrique Astuhuaman Davila, Daniel B. Hier, and Tayo Obafemi-Ajayi. I'm pleased to welcome one of the authors, Tony here, who is joining us today to talk about the work behind this award-winning research, what inspired it, the key takeaways, and where it might lead in the future. Before we start, so I'll give a very short intro about Tony. Tony is a graduate assistant in the Department of Physics, Astronomy, and Material Science at Missouri State University. His research focuses on explainable AI models in bioinformatics and drug discovery, combining techniques from machine learning and generative models. Tony, a big congratulations to you and your co-authors on this recognition. And thank you for joining us today. We are looking forward to learning more about your work.
Thank you. Pleasure to be here.
So let's start with the very basics. Tony, could you tell us what your paper is about and share the main idea behind it?
Yeah. So to start with, the original idea for this project came from Dr. Hier, who suggested looking at how centroids could make scatter plots more interpretable. At the CLS lab, the lab that I used to work as an undergrad researcher, the group had a strong focus on explainable models for biomedical applications. We together we discussed the concept that, and I was very interested in pursuing it as my college honors research project that I took as an undergrad. The main challenge is that high dimensional biomedical data, it's very hard to interpret when reduced to 2 or 3 dimensionals data. So you have often end up with scatter plots that shows clusters, but without much guidance on how or why the clusters are there in the scatter plots. So my contribution on this paper and this research was to extend the idea of combining the class centroids with feature centroids. That way the plot not only just summarize where the disease groups are, but also connect them back to the most important features or symptoms that define them. This made the results much easier to interpret and align with the lab's broader focus on explainability.
Okay. Thanks for explaining that, Tony. That really helped us understand your work better. Following that, perhaps you already explained a little bit, but I would like to ask or extend it even more. What made you interested in working on the explainability of high dimensional data? And how does your research help tackle that challenges?
In college, as an undergrad, we, as I mentioned before, Dr. Here was the professor that I used to collaborate alongside with Dr. T, that had this idea of centroids and that can make the scatter plots more interpretable. So, as a matter of fact, in the paper, we also used balance the classes sizes with a smooth algorithm. auto-select the dimensional reduction parameters using silhouette index, and use ensemble feature importance step like these explainable models like SHAP, random forest, and permutation importance. So we created this pipeline so that you can put like a data set for these to solve these complex biomedical, high dimensional biomedical data to choose which features overlaid over these centroids so that the plots stay both readable and principled.
Thank you, Tony, for explaining that. Now I would like to bring Professor Andrea into the conversation to get his thoughts and take this a bit deeper from an editorial and technical perspective.
Thank you, Sadia, and thank you, Tony, for your time. So I have a couple of questions. First question is, one of the main features of your work was combining class and feature centroids. How this allowed for improving the interpretability of two-dimensional scatter plots?
Yeah, that's a good question. So when you reduce high dimensional data into 2D or two dimensions, you get scatter plots that can look nice, but they're often abstract to interpret. So adding a class centroids helps this to simplify this visualization by showing the center for each group instead of just the cluster. But it still doesn't explain the features, what features are behind each separation, especially in biomedical data. So when we add the feature centroids, we give the plot biological landmarks. So, for example, in our neurogenetic disease dataset that we used, we saw that Charcot-Marie tooth disease line up closely with features like weakness, atrophy, and hyperreflexia. So the clinicians like immediately recognize those symptoms, so that the visualization now tells a clear story behind it, like to interpret it. So instead of just colored clusters, the scatter plot becomes more kind of like a map where the relationship between diseases and its own features is more visible to the human eye. And that combination of the class and feature centers is what makes the plots truly interpretable. Mhm.
Such a cool idea. Thank you so much. I have another question. Looking more broadly, how do you see the use of class and feature centroids contributing to the advancement of computational approaches in biology and general also in medicine?
So looking more broadly on the bigger picture, centroids actually give us a way to connect these machine learning outputs with human reasoning, right? So that humans can interpret it. So that's really important in these fields of biology and medicine, where the doctors and researchers need to understand why the models produce the results that they do, right? It's not only just a black box, but they get knowledge about these outputs. So accuracy in these models is not enough, but interpretability builds trust on these fields. So by showing which features are the closest to each disease group, our method helps to highlight the biological signals driving the clustering. This has potential applications, for example, in disease subtyping, biomarker discovery, and precision medicine. So it can guide researchers towards more meaningful associations. And at the same time, it can make the results more transparent enough for clinicians to validate it. So in a way, this approach of centroids are kind of like a bridge between computational biology and practical biomedical insights.
Thank you so much. I completely agree that interpretability is a key point, especially in clinical settings and also in biological settings. Thank you so much.
So, with that, we are going towards the ending of this podcast. So thank you, Tony, for sharing your insights. And thanks to Professor Andrea for being part of this conversation. Big congratulations again to Tony and the co-authors on receiving the Best Paper Award. And we wish you the very best of luck. Thank you.
Thank
Goodbye.
you. Thank you so much, guys. Thank you. Goodbye.